Close your eyes and imagine having a pair of molecular scissors, so precise, so intelligent, that they could edit the very code of life itself. Tonight, we're going to explore one of the most revolutionary discoveries in the history of science. CRISPR-Cas, a tool so powerful that it can rewrite DNA with the precision of a word processor, editing genes like you might edit a sentence. But this isn't science fiction. This is real, and it's happening right now in laboratories around the world. What makes this story even more remarkable is that CRISPR wasn't invented by scientists. It was discovered in nature, in the humblest of creatures, bacteria. These tiny organisms had been using this genetic editing system for billions of years as their own immune defense. Tonight, we'll journey from ancient bacterial battlefields to cutting-edge medical treatments and explore how understanding nature's own toolkit has given us the power to potentially cure genetic diseases, improve crops, and even bring back extinct species. So settle in and prepare to discover how the smallest creatures on Earth taught us to rewrite the book of life. Our story begins in 1987, when a Japanese scientist named Yoshizumi Ishino was studying bacteria and noticed something strange. He found unusual repetitive DNA sequences in bacterial genomes that didn't seem to have any obvious purpose. These mysterious sequences were scattered throughout bacterial DNA, like strange genetic bookmarks. Ishino couldn't have known that he had just discovered the foundation of what would become the most powerful gene editing tool in human history. For years, these sequences remained a scientific puzzle. Then in 1993, researchers studying Mycobacterium tuberculosis found similar patterns. More and more scientists began finding these clustered repeats in different bacterial species all around the world. But the real breakthrough came in 2002, when Dutch scientist Ruud Janssen coined the term CRISPR, clustered, regularly interspaced, short palindromic repeats. Finally, these mysterious sequences had a name, but their function remained a mystery. The puzzle began to unravel when scientists realized these sequences weren't random. They were memories. Bacteria, it turned out, had evolved a sophisticated immune system that could remember viral attackers and defend against them if they returned. Here's how it works. When a virus, called a bacteriophage, attacks a bacterium and the bacterium survives, it takes a small piece of the viral DNA and stores it in its CRISPR library. This viral fragment becomes a molecular wanted poster filed away for future reference. The bacteria also produce guardian proteins called Cas proteins, CRISPR-associated proteins that patrol the cell carrying copies of these viral fingerprints. If the same virus ever attacks again, the Cas proteins recognize it immediately, bind to its DNA, and cut it into harmless pieces. For millions of years, this bacterial immune system worked perfectly, protecting microbes from viral invasion. But in the early 21st century, human scientists began to wonder, could we reprogram this natural defense system for our own purposes? The race to understand CRISPR intensified in the 2000s. In 2005, scientists discovered that CRISPR gene families were present in most bacteria and many archaea, suggesting this was a fundamental biological defense mechanism. The breakthrough moment came in 2008, when researchers finally proved that CRISPR was indeed a bacterial immune system. They showed that bacteria could acquire immunity by incorporating viral DNA sequences into their CRISPR arrays and later defend against those same viruses. But the most revolutionary discovery happened in 2012 when Jennifer Doudna at UC Berkeley and Emmanuelle Charpentier, then in Sweden, working together, published their game-changing research. They had figured out how to reprogram the CRISPR system. Doudna and Charpentier simplified the natural system, creating a programmable molecular machine that could be directed to cut any DNA sequence scientists wanted. Instead of defending against viruses, CRISPR could now edit genes with unprecedented precision. Their insight was brilliant. They realized that by changing the guide RNA, the molecular GPS, that tells Cas proteins where to cut, they could target any genetic sequence in any organism. It was like reprogramming a molecular security guard with entirely new, wanted posters. The scientific community immediately recognized the revolutionary potential. Within months, laboratories worldwide were adapting the CRISPR-Cas9 system for their own research. What had taken nature millions of years to evolve, humans had learned to control in just a few years. For their groundbreaking work, Doudna and Charpentier were awarded the Nobel Prize in Chemistry in 2020, recognizing CRISPR as one of the most important scientific discoveries of the modern era. Let's take a gentle journey into the cell to see how CRISPR works. Imagine you want to correct a single letter in a book that's 3 billion letters long. This is essentially what CRISPR can do in the human genome. The process begins when scientists design a guide RNA that perfectly matches the DNA sequence they want to target. This guide RNA is like a molecular bloodhound programmed to seek out one specific genetic address among billions of possibilities. The guide RNA teams up with the Cas9 protein, forming a molecular complex that travels through the cell, scanning DNA strands like a spell checker, reading through a massive document, looking for one specific sequence. When the guide RNA finds its perfect match, it binds to the target DNA, like a key fitting into its specific lock. This binding signals the Cas9 protein to unfold its molecular scissors and make a precise double-stranded cut in the DNA. Here's where the real magic happens. Once the DNA is cut, the cell's natural repair systems rush to fix the break. Scientists can simply let the cell repair itself, which often turns off a problematic gene. Or they can provide a new piece of DNA, a genetic patch, that the cell will incorporate during repair. This second option is like using find and replace in a word processor. Scientists can remove a disease-causing mutation and replace it with the healthy version of the gene. They can delete problematic sequences, insert beneficial ones, or even add entirely new functions to cells. The entire process happens at the molecular level with precision that was unimaginable just decades ago. And because the system evolved in nature, it works in harmony with the cell's existing machinery, making it remarkably efficient and relatively safe. The medical applications of CRISPR technology are transforming how we think about treating genetic diseases. For the first time, we have a tool that can potentially correct genetic errors at their source rather than just managing symptoms. Sickle cell disease, a painful genetic condition affecting millions worldwide, has become one of CRISPR's first major success stories. In clinical trials, scientists are using CRISPR to edit patients' bone marrow cells, correcting the genetic mutation that causes the characteristic sickled shape of red blood cells. The process is elegantly simple. Doctors remove bone marrow cells from the patient, use CRISPR to correct the faulty gene in the laboratory, then return the edited cells to the patient's body. These corrected cells begin producing healthy red blood cells, potentially curing a disease that has plagued humanity for millennia. Scientists are also exploring CRISPR treatments for inherited blindness, muscular dystrophy, and even some forms of cancer. In each case, the approach is beautifully direct. Find the genetic cause and fix it with molecular precision. Perhaps most exciting are the possibilities for treating previously incurable conditions: Huntington's disease, cystic fibrosis, and certain forms of inherited deafness, all caused by specific genetic mutations, are potential targets for CRISPR therapy. The technology is also revolutionizing cancer treatment. Scientists can edit immune cells to better recognize and attack cancer cells. These edited immune cells become supercharged cancer fighters, programmed specifically for each patient's unique tumor. But medical applications extend beyond treating disease. Researchers are exploring how CRISPR might enhance human capabilities, improve disease resistance, or even slow the aging process. Though these applications remain largely experimental, they represent humanity's growing ability to take control of our own evolution. CRISPR isn't just revolutionizing medicine, it's also transforming how we grow food. As our global population approaches 8 billion, and climate change threatens traditional agriculture, we need crops that can thrive in challenging conditions. Scientists are using CRISPR to develop crops that resist drought, tolerate salty soils, and produce higher yields. Unlike traditional genetic modification, which often introduces genes from other species, CRISPR can make precise edits within a plant's existing genome. Imagine tomatoes that stay fresh longer, wheat that resists fungal diseases, or rice that produces more vitamin A to prevent blindness in developing countries. These aren't distant possibilities. They're being developed right now in laboratories around the world. The beauty of CRISPR-edited crops is that many are indistinguishable from varieties that might arise through natural mutation or traditional breeding. The technology simply accelerates a process that would otherwise take decades or centuries. With CRISPR's incredible power comes equally important ethical questions. The ability to edit genes, especially in human embryos, raises profound questions about the future of our species. Most scientists agree that using CRISPR to treat serious diseases in individual patients is not only acceptable, but morally imperative. However, editing genes in embryos, which would pass changes to future generations, remains highly controversial. The scientific community has established careful guidelines, distinguishing between therapeutic editing, which treats disease, and enhancement editing, which might improve normal human capabilities. Most research focuses on the therapeutic applications, where the benefits clearly outweigh the risks. International cooperation and thoughtful regulation ensure that this powerful technology is developed responsibly for the benefit of all humanity. The goal is to harness CRISPR's potential while preserving human dignity and genetic diversity. Perhaps most remarkably, we're still learning from the bacteria that invented this system. New CRISPR proteins discovered in different bacterial species expand our toolkit, giving us even more options for precise genetic editing.
Tonight, as you prepare for peaceful sleep, remember that you live in an extraordinary time. Humanity has learned to read, and now to rewrite the fundamental code of life itself. The same microscopic bacterial defense system that evolved billions of years ago is now helping us cure diseases, feed the world, and explore the very nature of what it means to be alive. Sweet dreams, knowing that the tiniest creatures on our planet have taught us some of life's greatest lessons and given us tools to build a healthier, more sustainable future for all.